I've known him for quite a long time, ever since he joined IISc Bangalore. He's a professor of chemical engineering at IISc Bangalore, and he has a BTech from IIT Bombay and an MS and PhD from University of Illinois Urbana-Champaign. He did his postdoc in theoretical biology at Los Alamos before joining IISc. His therapeutic engineering lab focuses on engineering of treatments and establishment of principles of the design of novel drugs and vaccines by developing mathematical models and simulation of the dynamics of infections. I have known Karthik also. Fine. Conference has been great. I've enjoyed every moment of it. Let me take the and Venkat and the other. Well, for being around for the last session. Hopefully, it'll be as exciting to you as the previous session. About an algorithm that we've constructed. Communities, and by structures, I mean the species that are present and the relative abundances that especially exist in large microbiomes, which make it somewhat challenging to predict. And I'll tell you what the challenges are and how we have addressed them. So this work we have done in collaboration with people at Unilever. Most of the work was done by my master's student, Amir. He's now gone off to the US to do his PhD. And then collaborators at Unilever, Jannavi Raut and Yugandhar Reddy. And so we're grateful to them for their contributions. Most of the modeling work was done at IISc, and the experimental components that I will talk about were all done at Unilever. So let me get started. Here's the question. How do we predict the structure of microbial communities, structures of microbial communities? For example, let's say the gut microbiome, you have a bunch of species there. What are those species and what are their compositions? What are the relative abundances? And how would that change when they are subjected to alterations in the environment? For example, one could think of a diet, and I jokingly say, this is the interesting cases that come about when somebody is subjected to an altered diet in a different environment for extended periods of time, and what then happens to this microbial community composition. So these are the sorts of questions that we are after. And the challenge here comes up because when you have a large number of these species present, then you can have what are called high order interactions, and they make the prediction of these community structures challenging. So here's the brief outline of my presentation today. I will set the stage up using a synthetic oral microbiome, which colleagues at Unilever put together, and bring about this notion of high order interactions. Based on that, you will appreciate the challenge that is involved in predicting the structures of these microbial communities. And I will present our approach to addressing this question, which involves this algorithm, which we have termed EPICS. And again, here I must thank Karthik, because he's the one who prodded us to come up with what he calls a cool name for a technique, right? And so hopefully this will stick. But EPIC stands for effective pairwise interactions for predicting community structure. And then I'll talk about how this does in comparison with experiments. And I'll talk about specifically two experimental examples, one from the literature and one that our collaborators at Unilever set up. This is the first part of my talk. Let's look at the synthetic oral microbiome and see what these high order interactions are. The colleagues set up a 7-member oral microbial community. This, they believed, was representative of several of the pathological situations that our oral cavities experience, including things like dental caries and periodontitis and so on. Here are the names. I'm probably not going to read them: Streptococcus sanguinis, SS, etc. I'm going to look at the abbreviations because those we are going to use as we go along in figuring out what the structures of these microbial communities are. These are the experiments that they did. They're, of course, interested in figuring out what the structure is of the microbial community that consists of all of those 7 microbial species, and what happens to that structure when there is some alteration. For example,